drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. If you like to read like I do, then you should check out the ReadWise app. It's totally free to download, and if you go through the link in the episode description or on my support page, you can get a free 60-day trial for zero cost. ReadWise is a really cool app that allows you to import digital highlights from over 30 different platforms such as Kindle, iBooks, PDFs, and more. It makes it really easy for you to review and find and share excerpts and quotes from whatever it is that you're reading, whether it's PDFs of scientific papers or books that you're reading on your Kindle, and you can even save and use highlights from physical books. I read a lot of physical books before bed, for example, or when I'm traveling, and the ReadWise app actually integrates with the camera on your phone so that you can take a photo of a page that you're reading, highlight a specific chunk of text from that page, and export it to the app so that you can reference it or find it or share it later. They even allow you to export really cool looking graphics of some of the highlights that you have in the books or the papers you're reading so that you can share them in an engaging way. So check out the ReadWise app today if you are a reader and you like to reference and organize what you read. Go through the link in the episode description or on my support page or in my free weekly newsletter at mindandmatter.substack.com and try the ReadWise app 60 days free today. Gyorgy Buzsaki, thank you for joining me.
Welcome.
Do you want to just take a moment to introduce yourself and talk a little bit about your background and what your lab studies?
I'm Gyorgy Buzsaki. I'm a professor at NYU. My interest is in big words, how memories are formed, how it relates to various things such as sleep, how sleep affects many aspects of life, from emotions to memory. I would put myself into a smaller category when I say I'm very much interested in circuit functions, the role of oscillations, various rhythms, brain-body interactions. So these are the key words that tie together everything that we do. The key structure that we are investigating is the hippocampus, and we reach out from the hippocampus to multiple areas of the brain. I've been doing this for a while. I think the one summary sentence is we would like to understand how cognition arises from action.
And when we think about the hippocampus, the stereotype of the hippocampus is that it's involved in memory formation and memory consolidation. It's a structure fairly deep within the brain. And when you learn about it in school or a textbook, it's typically pretty much just talked about in the context of memory, but taking a broader view based on sort of the full scope of all the research that's been done by your lab and others over the years, how would you start to talk about what the hippocampus's core function is? Is it strictly related to memory, or is it doing something more multifaceted?
Well, that's an interesting statement what you did, because actually most people who you ask in neuroscience or even in high school, what the hippocampus does, the answer is that it's the GPS of the brain. So it's related to spatial navigation. And these two thoughts of ideas have been running in parallel with each other. if you go back a little bit earlier in time, then the hippocampus was a regulator of endocrine functions.
Hmm.
There is a famous picture of Ramon y Cajal, a wiring diagram of the hippocampus that shows uh, how the different areas interact with each other, and he interpreted his observations with arrows, and those arrows all come from the entorhinal cortex, that is from the neocortex, and they all point downward to the fimbria, which is the output, which back then was thought to be going to the hypothalamus, which is also true now. And that gave rise to the idea that the hippocampus is a funnel into which the neocortical information is poured in, and then what comes out is the emotional control. So this is how the hippocampus became part of the emotional brain. Now this was going on for a while until the patient HM and until the discovery of the place cells. So once the place cells were discovered, 10 years later after HM, then two parallel pathways occurred in neuroscience. One was hippocampus is about spatial navigation, and the other one was it's about memory. And now we have to confront these two things, so bring together the two areas of research. And in a short nutshell, our solution is that probably the same algorithm is being involved in both memory as well as spatial navigation. Evolution made it possible that with the help of external cues, the animal can find around itself in the environment. And this is the real travel in the physical world. There is a process that I call internalization. So when you add many loops to the primitive brains, then the brain becomes more and more intelligent and is capable of making predictions on a longer time scale, can make a lot more things, and can disengage from the rest of the world. And this disengagement allowed the hippocampus as a navigational device to do internal navigation, which we call, not we, but I mean, it was Enver Tulvig who called it mental travel or mental time travel. So we can go back to the past and go into the future, but the fundamental computation that is going in the hippocampus is actually the same as in physical spatial navigation.
So it sounds like the arc of history here in research was that there were sort of two lineages of thinking. The first was the one that you mentioned initially. So this was rooted very much in anatomy. If you just look at the anatomy of the brain, there's lots of inputs to the hippocampus from the entorhinal cortex. So we think of the neocortex as sort of the higher brain where lots of the fancy stuff is happening. That's in essence anatomically pouring into the hippocampus. And then there's some connectivity there to the hypothalamus. And because the hypothalamus is what we think of as the endocrine regulator and a key emotional center of the brain, they're developed this idea that the hippocampus was sort of taking highly processed information from, let's just say, the thinking part of the brain or the higher brain, and using that to regulate what the emotional or endocrine part of the brain, like the hypothalamus, was going to output to the rest of the body. And then later, in parallel, so someone had a surgical removal of a set that included the hippocampus, and this implicated the hippocampus in memory.
And all of a sudden, it was discovered because of this surgical bilateral removal of the hippocampus that, wait a second, there is a problem. It's not only emotions that are going to be affected. It's not only endocrine function that will be affected. It is, in fact, one of the most fundamental things of a human being that was removed, which is learning.
So one group of people was thinking about things like spatial navigation. And we know that the hippocampus and other areas are very much involved in that. We could talk about place cells and grid cells, but obviously animals have to move their bodies through space. And these circuits seem to be very important for doing that, moving the actual animal's body through space. And there's this other thread of thinking that has to do with some other stuff, but to do with memory. But basically what you said was that somehow over the course of evolution, as more sophisticated and intelligent animals evolved, this circuitry acquired the ability to do essentially mental travel. So the same circuits that were used in actual travel through physical space allowed us to, say, think about the past and the future and do this kind of cognitive mental time travel.
So this is the picture until, let's say, 2024.
Mm. And then what happens?
Then we are in 2025. So we can think even in a bigger picture. The first question you ask, what happens in the hippocampus? What kind of computation happens in the structure? And over the years, we kept looking at various things and came to the conclusion that the hippocampus is kind of blind to modalities. It's not about that the animal is there is one thing that I forget to mention, of course, is that it's not about space only, but it's about time as well.
Mm-hmm.
And that was the era that was introduced by my good friend Howard Eichenbaum, who said the hippocampal cells that have place cells, that are place cells, that are also time cells. In fact, there was a back and forth for a while in the figuring out what fraction of them are place cells, what fraction of them are time cells, and so on. When it seemed to me that 100% of them are place cells, 100% of them are time cells, depending on how you address the question. And in the typical experiments that we designed are testing a hypothesis which is typically yours. We are making up a straw man, the hypothesis, and then we reject that. Now, I think it was forgot who was that person who originally said that he who knows only one side of the case knows very little, which implies that in every single experiment, you should test at least your hypothesis and the competing hypothesis,
Yeah.
or if preferable, 5 hypotheses, all the hypotheses combined. So this is what we have done recently, based or driven by the idea that there are so many ways the hippocampus is involved in many behaviors. So we designed the experiment. The very short summary of that, it turned out that the hippocampal cells don't respond to tone. That is not a auditory landscape. The hippocampus is about space only, not about time only. The whole thing seems to be expressed with words that I don't like either, but I don't know how to say, is that action planning is the goal of the hippocampal computation.
Mm-hmm.
And what was we designed this experiment that multiple hypotheses were confronted, and then we checked out 1, 2, 3, 4, 5 of the many possibilities. And what was left indeed is that action plans are what drive the hippocampal neuronal activity, and therefore hippocampal computation. And the way how I can respond to your question about spatial memory, for example, is that the big idea, and we can go back and talk about the even bigger picture for that, is that the hippocampus forms a representation of the external world, and the external world is a space-time configuration of Newtonian physics. You can revisit this.
Mm-hmm.
And that those representations are expressed to and available for the experimenter to relate it to time and relate it to space, as if really the brain is about reflecting the reality out there. So this is one idea. Our experiments suggest quite the opposite. Said, no. Brains come with a preconfigured dynamic, with lots of constraints. And this determines how the brain that is embodied into a actuator, which is the body, and which is placed in its own niche, that is the immediate environment with which the organism interacts with, is interpreted. So it's not a reflection of the world. but it's kind of a searching mechanism and say, given my constraints of the computational ability of the brain,
Hmm.
how can I interpret it, what I found out there?
Hmm.
Can you see the difference?
Yes. So with the latter idea, if I'm understanding you, you said pre-configuration, which seems important. It's not that the a representation of whatever external world might be out there can be made with high fidelity, but there are certain pre-configured constraints that probably have to do with the evolutionary history of that organism. Certain sort of assumptions are baked in.
Exactly. So when you said that, initial ideas were driven by the anatomy, that's true. But again, then you have to ask, what anatomy?
Mm-hmm.
Whose brains are we talking about?
Mm.
We are talking about the mammalian brain, where as you very eloquently summarized, much better than I said, that the intelligence from coming from the neocortex goes to the hippocampus and so on. It goes to the endocrine control system. Uh, but if you look at a simpler brain, such as a bird or a lizard, or even simpler, then you can see that the hippocampal primordium exists, but it communicates not necessarily with the neocortex, which doesn't exist. Then the wolves, then the corresponding structure that we call neocortex is tiny, teeny.
Mm-hmm.
So all of a sudden, you see a different animal and you have a different view. Also, when you say, okay, spatial representation, what is the representation of space of a bird that flies, or a bat that flies in 3 dimension, or a fish that is flying, that's lying on the on the bottom of the ocean all the time,
Yeah.
and and the human that is moving mostly in 2 dimension. So our perceptual world, our truth that we learn about the world seems to be very different depending on how the brain constrains the kind of way how you can imagine, how you can construct the world that surrounds you.
Mm-hmm. So a few different things we can start to talk about here and tie together. So the hippocampus. So just tell me if this is how you start to think about the hippocampus. So the hippocampus is coming up with a representation of the world that is constrained in certain ways, and it's constrained differently in different species, but it's a fairly amodal or fairly abstract representation of possibilities. It's not tied to one sensory modality in the way that, say, we've got the visual system, which is mapping out essentially the retina and how it relates to the external world. And we've got other sensory modalities, and those things eventually get integrated together. But the hippocampus doesn't care just about one modality. It cares about the space of potential actions that could be performed given what the animal is physically capable of.
Almost correct. We can go one step further, being more radical, and say the hippocampus doesn't really have to represent anything.
Mm.
Doesn't have to have a picture of the world.
Hmm.
When when I think about, after this interview with you, uh, of course, the beard is part of you, the earphones are part of you, and so on. But in reality, when you think about your best friend or anybody, you don't have a picture.
There's a feeling.
There's a abstraction of that thing. And
Mm-hmm.
when you say, "Oh, you know what I'm talking about. This is a table," and then you can think of close to the last table, the most favorite table or most known table, but you don't see a table. So there is no such a thing as a representation of a physical object in its reality.
Mm-hmm.
And that's a very important distinction, because if you follow the vision research, the vision research, or any kind of sensory research, we are trying to reconstruct how the brain represents the things that are out there. But it's quite naive, I would say. Now, when you go to the hippocampus, which is in the middle of the brain, and it doesn't have a modality specific transformation of things there, that is even more remote. So why do we need the word representation. This comes from the what I call the outside in view of the brain of neuroscience, which is that initially we were thinking about, not we didn't care about the brain 2000 years ago or 4000 years ago, but we cared about the spirits and we cared about the soul, and we cared about later the mind and so on. So we wondered, how spirits are made, how the soul is made, and why we have it. And then there were answers. People came together, and we were thinking, and they said, well, the soul was given to us, and depending on which society lived in. If you lived in a Christian society, that of course the answer was God gave it to you. But it's very similar in many other religions, namely that you are given this favor, given this present, and you have to use it wisely.
And that sort of, if we take those historical ideas which show up in the context of religion and we tie it to what you were saying earlier, what we're really talking about here, I think, and people have written about this from the historical perspective, from the psychological perspective, and if I'm hearing you correctly, maybe you have a neuroscientific perspective on this, but this ties into what you said earlier around preconfigured constraints. We are given certain constraints based on our evolutionary history, and these tie into things that are just givens for what our brain can do and how it can represent.
Yes. But in the religious case, it was given to you. It's not the brain and preconfigured; it's the soul, and it is the soul itself that is given the ability to choose, and it was given to you to discover the beauty that was given to you. And if you are going in the right direction and you always choose good, then you will be rewarded in your afterlife. If you are doing the wrong thing, then you are being punished in the afterlife. So that's an interesting simplification of thinking, but this is what I sort of trace back in the history of the ancient history of neuroscience. So thinking about the brain is that indeed that is the way how it was created, that we are perceiving those beauties or the outside the world because it's objective, and then we are given the choice, a decision process that is connected to free will and all these complicated things. And then at the end, there will be an action. This is the outside in idea. But then the problem was always in the middle, which is the free will, the decision making. You can call it black box, or whatever you want to call it. It is a thing that is very difficult to address. Okay. Now, of course, this idea came to philosophy, British empiricism. That was my next step, and then psychology, especially many codified summaries of this. And then when people were trying to do something in the brain, then they said, "Oh, what do we want to find?" And they wanted to find answers to all these preconceived ideas that were concocted by humans 2,000-3,000 years earlier. And then many words such as memory, space, time, decisions, and so on. We thought that these are the categories that really are important because we inherited them, and you can find these categories in William James's, 2-volume fantastic books. And the goal of neuroscience is basically identify homes for each of these chapters of James' book, and find the mechanisms and the boundaries in the brain with the same boundaries that have been conceived by people who didn't care or didn't even know that all these complicated things happen in the brain. Now, this is obviously an exaggeration, what I'm saying, and it's obviously a cartoonistic way of thinking, but there is no real competing theory out there or competing formulation of the problems. How we arrived to the situation where we are at now, or we were in the past decade or so. So this is where you can say, oh, this is the frame. This is the framework. We have the framework, and we just have to move forward. And then you come to the hippocampus and then you start taking seriously these words. You said, yes, if this is the framework, and then we have to find definition, we have to find agreements among humans. And then once we have this consensus, then we can go and deliver mechanisms that are explaining what we want to say. This is that we find explananda to the explanandum, pretending that we already know what needs to be explained.
Mm.
And if it, I used to say jokingly that if we don't understand something, we can make up a few words and pretend that those words explains the things that we don't understand. Now, we were given many of these words, information. I have no clue
Yeah.
what information is. Processing, that's another thing that is very difficult to say. And many of these decision making and so on, these are very difficult concepts that we take for granted and explain.
Mm.
Then I focused in eventually on two key aspects of these words, space and time.
Mm-hmm.
Now, space and time are the most important things in human thinking because these two things give you a coordinate system for our thinking.
Mm-hmm.
Everything we do, everything we know happens somewhere and sometime. And that's an easy explanation. But when you, and of course, this is how we relate everything to the hippocampus, is that we can say, oh, the hippocampus has memories, it has spatial navigation abilities, and we navigate in space. Then we remember something that also has to be placed into space and time. And then we find these cells that behave like place cells. They behave like time cells. But the same neurons also tell you about the content.
Hmm.
So making all these complicated things a little bit more simplified, we come to, not we, my smarter people than us today, such as Enver Tulving and others, they say we have a definition for memories. Let's start with what kind of memories are important. And of course, memories are pretty complicated, but let's say there are memories that are declarable, that I can tell you with words. And there are things that I can't tell, but I know I remember how to ride a bicycle. And so these are two major things. One is that are consciously declarable, and those are that not. Now, this is an interesting approach because it again starts with a chauvinistic, human, language-centered point.
Yeah. Before we go on, so I think what you're saying is, if I sort of compress much of what you just said, when we talk about stuff, we have to use words. We have ideas around memory, we have ideas around space and time, but very often, even very educated people, even neuroscientists who study this stuff, we don't stop to reflect on the fact that these ideas have been around for thousands of years. People have had a conception of memories and space and time and all these things. And we've inherited these ideas down the millennia. And there are certain sort of conceptual assumptions baked into a lot of things. And some of those can start to break down when you start to look at the details of what in the hippocampus or the brain is doing. And it comes from the fact that people often haven't reflected on the ideas we've inherited. We sort of assume there's going to be a memory place and a spatial navigation place and a time place in the brain, and these things will tie together in a particular way. But then you and others start to make observations in the brain and the hippocampus that start to make you think, well, actually, things aren't so neatly parsable according to the ideas that we've all just been assuming because we've inherited them.
Mm-hmm. And of course, I'm not saying this is specific for neuroscience, not at all.
Yeah, it happens. It happens a lot in science, in the history of science. You mentioned how we don't understand something. We can just make up words and fool ourselves into thinking we understand it. You can think of many examples in history, like phlogiston is the example of where fire comes from, or the luminiferous ether. Like we've seen this happen many, many times.
Exactly. Exactly. So this is the time perhaps where we are in the evolution of neuroscience or brain science, where physics was, a century earlier, or genetics was also a century earlier. And then people, when we try to understand mechanism, and we try to understand the details, and then we see that the facts go against each other.
Yeah.
And then we have many more facts that are uh, they don't help because the framework is the problem.
Hmm.
Then you start to change the framework.
Hmm.
And I think neuroscience is facing a similar crisis, which is a good crisis in the sense that now we are questioning those ideas. So just I used a couple of examples because we said we try to understand the brain, even the very specific things such as memory from the human language point of view.
Hmm.
So now this declarative memory idea is a great thing. Larry Squire is a very, very good friend of mine, and he is the one who will get credit for the declarative memory. But what do I do with this uh, consensus-based uh, uh, declaration when I'm studying memory in a rodent
Hmm.
who cannot communicate to me verbally anything? So then you say, well, what is the essence of that definition? And then you can think about either abandoning it or said, well, what episodic memory is interesting, and we can go discuss the episodic memory separately, is what happened to me and where and when. The key thing here is what is called the autonoeticness. The autonoetic, that is how I do feel about this. Now, this is very difficult to study in humans.
Hmm.
It's almost impossible in animals, especially in simple animals, because the autonoeticness is not easily measurable. It could be declared only. So then you can say, well, it's super important because it captures something important, namely that I am who I am because of my memories. And then you can say, well, what is important here is the individuality. This is the essence of individuality. But this is the nature of biology. Biology is defined by the fact that there are individual organisms and which are not dividable, and their action is always territory oriented. They want to define themselves as different from the others. And there are exceptions, but most of the time, this is what happens there. Said, oh, then of course it makes sense to talk about a agent that is capable of getting around by its internal actions,
Hmm.
and we call it self or egocentric. And then when you look at from the third point of view, when you are looking at others, then you can say allocentric. Now it just turns out, if you remember these two words, allocentric and egocentric, that there is allocentric navigation and egocentric navigation. And there is a 3 decades of history starting with John O'Keefe and well, I would say mostly John O'Keefe and later on many other people who said, oh, this is interesting. How do we make maps? How do we go from ego to allo? And which one is more fundamental and so on. And this is reflecting the whole discussion about the details of Gestalt psychology, whether we see the whole or we build up the whole from elements,
Hmm.
or from the wholes we break down the important things and we go to the specifics. And so that kind of approach went back and forth, but in parallel, this definition from the egocentric memory, which is the episodic memory, which is about me, and the other type of memory, which is semantic memory, which is also declarable. This is the other large category. And look at the relationship. And they said, but this is good. The way how we make semantic memories is we get many experiences that have something in common.
Mm. Yeah.
And the intersection of those commonalities are useful because you can subtract or or abstract all those things that led to that one thing. You forget about how you got there, the spatial temporal situations, and you just end up with that explicit thing. And this is the explicit or semantic memory. So just to give you an example, and it's a trivial example I always use, is that when you see the first dog in your life, that's an episode. That's a personal experience. You see another dog, and another dog, and then the spatial temporal conditions, how the experience occurred, are not so important. In fact, if you generalize, when a child sees a uh, uh, a, a lamb, he will say, "Oh, it's a dog," because there is no distinctive feature. And then the other things, they disappear, and then it becomes a dogness, which becomes a explicit memory. Now, the good news is that this is exactly how the other side of the field, spatial navigation, works. Because in order to generate a map, you have to have your own experiences. You have to visit every part of the world. Uh, and once you visited every part of the world, you can generate a map, you can draw a map with the advantage that now you can go from anywhere to anywhere else and taking routes that you have never taken. But first, you have to do the ego part, just like you have to do it with memory. And then you end up with place cells, and the place cells are the embodiment of abstraction because they are intersections of the XY coordinates, and they are just like semantic memories. So place fields are corresponding to semantic memories. To
Yeah,
make a long story short, it seems there is a nice parallel between egocentric navigation and egocentric episodic memories, and allocentric spatial navigation, and allocentric, this is individual, independent view of things, which is semantic memories. And the two are probably produced by the same algorithm.
The same algorithm could, in principle, be doing both things. What we think of as two things is really fundamentally one type of
One,
computation.
exactly.
So let's break this down a little bit for people and give them a sense for how you study this, and then help reinforce some of these ideas and make this a little bit more grokable for people. So let's just start with some common experimental setups in your world, or the world of hippocampus and memory and navigation. So we talked about place cells in the hippocampus. What are these, and what is the type of experimental configuration that they were discovered in?
So interpreting history is very difficult. One of my closest friends is John O'Keefe's in science. And I remember his first interpretation, how place cells were discovered, but his later interpretation is different. And I believe in the first story that he told me, which is that he was looking for place cells because of Tolman's ideas that animals are not automatons. They are not Skinnerian robots. They are not Pavlovian robots. But in fact, they have goals, and therefore they can relate themselves to made up goals. And in order to do that, they have to have he called it a map, initially a spatial map, but then he said it's not necessarily spatial, let's call it a cognitive map. And so John O'Keefe was looking for the instantiation of this in the brain. And he found it in the hippocampus. Now, the reason why I'm venturing to say that, and then the historians will figure out the details, is because if you go back to the discovery paper, the 1971 paper, I would say nobody but O'Keefe would conclude from those data that those firing patterns actually place fields. Because, again, please forgive me, John, but if you look at the figure details, there is nothing really tells you, and it's very difficult to observe that you put back the same animal at the same time, even though he was listening to the neurons, and so when the animal comes back to the same place, it fires again the same frequency. It's difficult to convince yourself that the animal was not doing some specific movement, or it was not looking at something
Uh,
specific. So there were
so, so okay,
no
so
good.
what you're saying is if you go back to the original place cell paper, so for those who don't know, you put a mouse or a rat in a maze, and there's different places spatially in the maze, and when it goes to the same spot, the same neuron fires in the same way. One way to interpret that is that it's firing because it's representing that place. But you're saying potentially, if you look at the same data, you could say, well, maybe, every time the rodent goes to this arm of the thing, it has a tendency to, make a certain movement because it's being provoked by something in the environment. And it's not actually a representation of space. It's a representation of some action taken at that place or something like that.
Exactly. So that mistake, quote unquote mistake, has been done repeatedly over and over again, that people have described place fields in any part of the brain, from locus coeruleus, from the VTA, the olfactory cortex, everywhere, because this sort of constellations of activities occur unless you do many, 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 many controls. So the
Yeah.
reason why that paper is a prime example of a reflection of an extraordinary mind, because despite the fact that almost nothing holds in that paper, everything holds in terms of the hypothesized function of those cells. And everything that predicted there was confirmed again and again and again. That was the vision. And the reason why the vision there, I think, was because this is such a rare thing in science, that you have a preconceived
Yeah.
idea and you actually find it.
Yeah.
Typically, we find somebody by, most of the time, we find things by
Yeah.
accident. We
Yeah,
find,
I love how you're talking about the history of thought behind all of these episodes in history. What you're saying is he really came into these experiments with a strong preconceived notion of how things must work, and he took that seriously despite alternative interpretations of the same data.
and it's so rare in science. It's so rare. And he made another fundamental discovery also. Again, it's my interpretation. He already had a preconceived idea. This was called the phase precession. He was looking for a pattern. He didn't have the computational abilities of today. He had a storage oscilloscope that he and a few of us had. And then he found one run and said, there it was. The place one, place two, place three, and there is phase one, phase two, phase three. Is there. And and so he's an extraordinary mind because he can look into the brain and find something that fits the right picture. In most of the time, it's the opposite. You know, we never find what we are looking for. Instead, we find something, and I forget who said that the most important thing in a science laboratory is to say, Richard Feynman probably, that it's funny. It doesn't fit. Let's investigate it because it seems to contradict everything I knew before. This is the typical entry to a big theory, rather than the other way around. Okay, so this was the kind of thing, and then it, of course, and again, a very unusual thing that it wouldn't happen easily today. That you have a half job, you don't have a lot of job security, and so on, and you decide to write a book based on this preliminary observation, and he had two or three more papers afterwards. But then the 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 explanatory power was so good, and his preparation for this was already there. So he wrote this Bible, and then of course there was a small group of people who followed him. Uh, you can see 2 decades passes, and there is only dozens of papers or so about spatial navigation. But then when things became a little bit better, especially with phase precession, I became interested in this whole problem, not because of space and time, which was too noisy for me, but because time and phase as a mechanism entered into the picture. Said, oh my God, it's an interesting thing. O'Keefe also discovered that it's not only the firing rate of the neurons, but the relationship between where the action potential occurs on the phase of the theta cycle in a 100 millisecond time window is telling the rest of the brain, or at least to the experimenter, where the animal is. So you take a 100 millisecond time snapshot of activity, and you can precisely tell where the animal is. And then it's an interesting thing that something inside the brain, inside the hippocampus, is organizing the activity at the time scale of tens of milliseconds that relate to the animal's actions at the several seconds time window. And I think this sort of findings, and that one after the other, is what made people attend to the hippocampus for perhaps one reason that I can summarize with this metaphor easily. For computational neuroscientists, noise is the crutch of everything. Without noise, nothing works. So something externally has to be given to a network to do anything useful. For me, it was the other way around. The brain, especially the hippocampus deep in the brain, is a self-organized structure. It generates patterns, but that's not noise. Your noise is my signal. And what I could say, rightly, I would say, is that the history of the hippocampus is the history of denoising the brain. The more you know about the details, the more neurons you record from, the better designed the experiments are. Every single action potentials can be accounted for. It's not that there's jitter and all these things. It's important because it's costly. Every action potential costs 10 to the ninth ATP. So when this picture started to be looking crispier and crispier, I said, wow, the hippocampus deep in the brain, so far away from the external world and external controls, the external control of humans, is giving rise to a explanatory power at such a detail that the visual cortex, the auditory cortex, and the sensory cortices are not capable of doing, then a lot more attention was paid, and it was an exponential process. The discovery of the grid cells, another extraordinary discovery that everybody scratched their head. So how on earth this dynamic comes about? Why is it there? And anybody who is a scientist is blessed by a high dose of curiosity and said, I never thought about that. And if I haven't thought about that and it's there, then maybe that structure or that dynamic tells us something that could be carried on or generalized to other areas of the brain. So this is how a noisy structure, that was about memory, navigation, and so on, all of a sudden became a circuit that gave rise to a explained dynamic. And at the same time, its anatomy and its microcircuitry was investigated in exquisite details. And one part of my life was figuring out the hippocampal interneurons and how the inhibition contributes to all of these interesting things that happen in the hippocampus. So everything combined this way gives you a feeling that, yes, it's coincidental occurrences of multiple events that gave rise to the prominence of the hippocampus in brain sciences.
You mentioned the energetic cost of action potentials. So neurons are very metabolically expensive. It takes a lot of ATP. It takes a lot of energy to do just one action potential. And obviously in the brain of a mammal, you've got millions or even billions of neurons. And so this is a very serious thing to consider. And when we start to think about things like information processing and memory encoding and all of the stuff that we're interested in uncovering in terms of what the brain is doing and how it's doing these things. You mentioned that, there's a very prominent concept out there in many neuroscientists' minds of noise, that there's a lot of spontaneous activity, that neurons are sort of leaky, they fire when they're not supposed to. But that's also a little bit awkward to think about if action potentials are so energetically expensive, and we just think about this from an evolutionary perspective, why would an organ like the brain that's that expensive evolve in such a way that you have all of these misfirings, all of this noise happening all the time? And can you say a little bit more about that? Because it started to sound like you were saying, no, maybe these are not misfirings. This is actually endogenous structured activity from self-organizing structures in the brain, and it's supposed to be there, and it's not just noise.
So this is a good direction, and you use the keyword information, the thing I don't understand. I don't think anybody does, but you can confront what information means to a neuroscientist and what information means to an information scientist. I used to be a radio ham and studied Morse code, and I knew a lot of things about coding in the simple world, and in every single system which requires messaging requires that the stuff that to be sent have to be packaged. If our conversation would be one very long sentence, it would be very difficult to decipher, but not impossible, because we've got many letters and many sounds in human use. But in the Morse code, there is only tida and silence, which is the end of the letter. In the brain, there is only a spike, and every pyramidal cell sounds the same. So then you say, what is what you call information, or how is it transmitted by spikes? And then you go back to the smart people of information theory, and you say, well, they already told you that one spike or one pulse is not information. The information could be only between between two. That is, you can call it the time elapsed, or what is the time jitter between the two. Now, if that is the case, then neurons that spike at a low rate, by this definition, cannot transmit information. Now, this is a good thing, because you can measure and how many spikes occur at a very long intervals. And we have done this. We have done a diligent homework with this, with multiple areas of the hippocampus, entorhinal cortex, prefrontal cortex, and many other cortical areas. And we asked simple that what is the difference when a neuron fires sporadically? And you can quantify it. And what is the mean firing rate of the neuron? And what if you look at the distribution of the mean firing rates over various time windows? And we came to the conclusion is that the mean firing rate distribution follows a log normal distribution, which means that the majority of the neurons are very, very, very silent. And only a minority of them are talkative and loud. And if you do a mean of the mean, then it's always the fast-firing minority that captures everything. It's the winner-take-all mechanism. And this is a dangerous thing to know, because today we are obsessed with things what we call dimensional reduction. This dimensional reduction is based basically by looking at, we do it the same thing. Of course, I'm not saying we are innocent, that we take 500 neurons and then we reduce it to a two-dimensional map. But we never ask the contribution of the individual neurons, but I know that it's only a small minority whose pattern is responsible for all of this, and we don't know about others.
Yeah. Okay. So if you record from 500 neurons and you make the assumption that they're all equally contributing information, but that's violated according to you.
Exactly. Well, John Hopfield just received the Nobel Prize, and he explicitly said, these are in this attractor networks, everybody is equal.
Hmm.
That's so far from reality, so far from reality, because that's not the case. So now, having said that, when you look at individual neurons, then you can say individual neurons fire like tick, nothing, and the brr, and so on. So we looked at it. What is the long interval, and what is the relationship with long intervals and short interval? The short of the long is that when a neuron fires at a higher frequency with short interspike interval, this is the time when it fires with other partners.
Hmm.
And it typically fires in a particular cycle that we call brain oscillations or brain rhythms. And when the neuron fires sporadically, this with a long interval, it has no partners. So now we can declare that there are neurons, there are action potentials that are used for information processing. These are the time when brr occurs. And there are times when there is just a spike. And the cute thing now that you can quantify this and ask how many, what fraction of neurons where in the brain are used for deliberate information transmitting or just making noise. And the number is a staggering 50 to 60%. 50 to 60% of the spike at least in a red brain, is useless, quote unquote, because it doesn't transmit information. So what is it doing? Well, what are they doing? And the answer is they maintain the dynamic of the brain.
Hmm. So not
Because
transmitting information, but that's puzzling because these are very metabolically expensive things to generate, and
exactly,
evolution would not create something that's that wasteful.
exactly. And so 60% of the waste should be useful for something. And our conclusion is that the most important function of the brain first is to maintain its dynamic. And this is exactly the case in what happens in an AI network or in a brain network is that you put in from external sources something that the brain can generate itself, but it's not noise. It's a resource that is needed that you maintain the activity. If you take away the noise from the network, from an artificial network, it is dead. And I assume if you would take away 6% of the spikes in the brain selectively for this non-communicating neurons, it would be dead because there would be probably nobody to drive the interneurons. Nobody would be able to give a little bit of a reminder for the mini that occur all the time in the brain. It would just look like the downstate of non-REM sleep, and everybody would be silent. Uh, Instead, as I said, this is a fundamental observation, I think. So we can say that, going back to the beginning of our conversation, said, well, brains were generated and they were given a task by evolution or by God, depending on what side you are. And they say the most important thing is to maintain its own dynamics. And then the second most thing for me is to generate an action. And the third one is to register the consequences of this action. This is what I call the inside out approach, as opposed to the outside in representational view that we discussed at
Mhm.
length.
And are you essentially saying that your view is that the primary thing is for the brain to maintain its own internal dynamics because that's a prerequisite for being able to generate actions and then learn from them?
Exactly. So the brain generates pattern after pattern after pattern. And again, if you would like to contrast the two worlds, I would say in one case you have the Hopfield type of approach that everybody is equal. We have Hebbian rules, and then with every single learning and experience, we adjust everything, and it's a complicated, very energetically expensive process. The other extreme is they say, no, the brain is it's like a Lego game. It comes with preconfigured dynamic and preconfigured sequential activity, and the sequences can be cut and concatenated easily by inhibition. And inhibition can do tons and tons of things that have been attributed to synaptic plasticity, which is energetically very costly because it requires calcium influx and so on. So, and then the learning becomes not a synthesis that you build up for every single time from scratch. But in my world, we have the patterns already inside the brain, and then actions that lead to something that useful for the organism will be linked or
You're
matched.
linking things that are already there. You're linking Lego blocks that are already there, to use your analogy, but you come pre-configured with a certain number and pattern of Lego blocks that can be put together in various combinations, but a finite number of combinations.
Exactly. So by my previous postdoc, Ken Harris, did a very beautiful article, already calculated this, that, for example, in the auditory cortex, the realm of possibilities, that is what patterns exist, is not infinite. It depends on the brain, and you can look at it during sleep, for example, because sleep is the honest expression of the brain. It doesn't lie, like we can do with when we are conscious. And then it said every single pattern that we observe when you evoke firing in the neurons with any frequency, any auditory pattern, falls within the range of these possibilities. And then another student, and there was a student of mine, who was at about the same time when we were discussing, going in the lab, when we were discussing the same kind of thing, George Dragoy, he went on and he looked at it, the hippocampus view, and he also showed that indeed there are patterns in the brain that can predict what happens to those neurons in a novel environment. And then this is the theme that we are working on ever since, that indeed, if you look at the brain this way, it's more economic and more easier to explain. And inhibition comes forward as a organizing force. And plasticity, as a very important thing, is not abused that much as it used to be.
Let's talk a little bit about inhibition rhythms and things like sleep and memory. So one of the ways I want to steer you here is you've done a lot of work on brain rhythms, a lot of work on the different phases of waking and sleep. And one of the things, we'll just sort of assume people are familiar with things like EEG here, and that obviously when you record EEG activity, you can record from large ensembles of neurons rather than individual neurons. And you can do something like EEG. And when you do that, when an animal's awake, when it's sleeping, when it's in non-REM sleep, when it's in REM sleep, you see different combinations of rhythms that define each of these brain states or behavioral states. How do you start to think about the relationship between those brain states, the rhythms they contain, and what those are allowing or disallowing when it comes to memory consolidation and recall?
What you said is absolutely right. You can find all sorts of rhythms. And again, history here is very interesting, because when people were talking about rhythms, they involved mostly people who looked at very simple organisms, which have a lot of rhythmicity, such as the gastric ganglia, uh, or locomotion. People who were obsessed about the human cognitive abilities, they looked at the EEG and they said everything that happens when you are doing something useful, which is cognition, gives you a flat EEG. Rhythms occur only when you are asleep or when you are epileptic. So that was the state of the art until 1970s. And when you use higher resolution electrodes and better designs and so on, and again, hippocampus is an interesting thing, because if you stick an electrode into the hippocampus, you cannot not see this huge data activity. It was actually discovered, at least in a freely moving animal, by my mentor, who was the first one who looked at how theta could be related to any behavior.
So theta activity, that's a particular brain rhythm that you see prominently in the hippocampus.
Yes, exactly. And that was a great lesson to me because my mentor was an extraordinary man, Andrei Grosjean, who looked at it and said, "Oh, this is related to the orienting response of the animal, which is a Pavlovian term, and it's a response." And there was another person at the other end of the world in Canada who became my postdoc mentor, who said the opposite. No, it says it's related to the voluntary action. And so one was the perception side, one was the
Yeah. So again, it's this clash between something being evoked from the outside versus something being present from the inside already.
exactly. So that was good thing. At the same time, I could tell you that I worked on the radio ham. I knew about amplitude modulation, frequency modulation, and so on. That I know they are very important carriers. Fast forward many years, it turns out that there were many rhythms that have different names, different mechanisms, but it looked to me like a periodic system that had a chart, and every range of the frequencies fell into its own spot if you plot it rightly, which is a logarithm scale rather than a linear scale. And today we can go and include not only the brain rhythms, but also the body rhythms, the autonomic nervous system, and the body rhythms. And we can go in this logarithm scale down to 24 hours. So that alone is interesting, right? That every measurable interval is represented by one or another brain oscillator or body oscillator. The more important thing is that all of these, from the very slow 24-hour rhythm to the fastest sharp wave ripples in the hippocampus, form a hierarchical system. What does it mean? What it means is that the phase of the slow oscillation modulates the amplitude of the faster one. And the phase of the faster one modulates the amplitude of the even faster one. So it's like, there's 24 hours in a day, but it has the moon cycles, and we have high tide and low tide. And that can be measured by looking at the water levels.
Mm-hmm.
And the water levels are phase modulated by the day. Because if your 24 hours is a cycle, is a sinus, then you can say, oh, 6 o'clock is a good time for surfing, and 12 o'clock is not a good time, but 6 o'clock in the evening is another good time, and so on. So this is a natural thing that happens in nature also, but it happens in the brain very, very powerfully. Okay, good. So this is a good thing that gives you a timing, but it became real science when we realized that at least the brain oscillations, they are all tied to inhibition.
Okay, and before we go further here, so we've got these different brain oscillations. They interact with each other in interesting ways that you're talking about amplitude modulation by one rhythm to another, and we can get more into that. But where do these rhythms come from? If are you saying so? Do these rhythms come from the intrinsic biophysical properties of certain neurons that make them just prone to firing at certain frequencies?
Those are very important contributors, but circuits are equally important. So, for example, in the heart, the intrinsic properties are more important because heart muscle cells don't communicate very well with each other, only the neighboring ones. There is no axon. So the H current in the heart is important. And there is such a thing as H current with the same time course also in many neurons, especially in thalamic neurons, but also in hippocampal neurons. So indeed, they give you a property that at this particular frequency, if you are hit harder, then you resonate.
Mm-hmm.
So resonance and oscillations are related things.
Yes.
One can do the amplification for the service of the other. But once you start lining up things, and connecting things, then you can have a sustained oscillation.
Mm.
So when you have a pyramidal and interneuron, for example, that this is a tug of war between inhibition and excitation, in every field of physics, the stability or balance is always achieved by oscillatory patterns. It's not flat. It's it's very difficult to do it like this. That everything is equal. It's always a little bit of out of balance, and one brings back the other. So that's a good thing. That's again an enormous energy savings here. Just imagine that you have to keep every pyramidal cell's membrane potential very close to the action potential threshold.
Mm-hmm.
That would require enormous resources. But when the membrane can fluctuate, and then when the things occur at the right time, then because it's already depolarized very close to the threshold, then the neuron can fire. And if it is can coordinated across two neurons, 5 pyramidal cells, hundreds of pyramidal cells, that can be done with inhibition, then things are coordinated already. So now the inhibition coordinates the activity of the neurons, but also terminates the messages. So these oscillations in the brain are called relaxation oscillators. That means that there is a separate sending and receiving path. So there are times when the neurons, pyramidal cells, fire mostly. In the hippocampus is at the trough of the theta cycle. This is when it's message that's sent out. And the other phases are there that the hippocampus can be perturbed. So this is a useful thing. And then you can now say that, unlike in the computer, unlike in other system, physical system, in the brain, the communication speed is slow because of the slow conductance of the axons. That means that time or phase is also related to space. If you have only a short period of time available, let's say 5 milliseconds of a ripple cycle, then the activity cannot spread far away. It remains local. But if you have 100 millisecond time, let's say during an upstate in the neocortex, then many neurons in wide areas of the cortex and hippocampus can fire together or can be recruited. So again, through this amplitude phase relationship, you can now say that, whenever computation occurs locally, the local computation is under the control of a global computation, which is a global oscillator. And the other way around is also true whenever there is a local computation that can be broadcasted globally. So these rhythms on oscillations gives you time windows within which neurons can come together for some purpose. I can give you an example for one purpose if you want.
Yes.
So one of the most prominent, not prominent, but mostly investigated patterns called gamma pattern or gamma oscillation. The duration of a gamma cycle is about 10 to 30 milliseconds. I said, why is it the case? The answer is that this is the time constant of the membrane of a typical pyramidal cell in the cortex. What does it mean? What it means is that a pyramidal cell can integrate the action potentials of many neurons, any number of neurons from anywhere.
In that window.
In that window. And if you happen to fire 50 milliseconds away or apart from the first action potential, then that you must belong to a different entity, a different cell assembly. So the goal, if you want to talk about goal, the goal of this gamma cycle is to bring together neurons for one purpose, discharge the target neuron. Because the target neuron, due to its membrane time constant, will integrate all the spikes that are coming into this time window. It just happens that this is the time window of spike time dependent plasticity for a good reason, probably. It just happens that this is the time window of the AMPA excitatory receptor and the GABA A receptor. So there are biophysical reasons, and there are these other reasons why this is a useful time window. Now, you can just go one step further and said every single gamma cycle is a cell assembly. So this assembly formulation of mine is very different from the Hebbian cell assembly. No, Hebbian cell assemblies have no time. And it's impossible to define what is an
if you're part of an assembly, you're always part of it. But what you're saying, in your view, is whether or not you're part of a given assembly depends on the timing of things.
This is a very pragmatic, effector-centered definition, whereas Hab's definition, with all my respect, is very loose. What is assembly? Assembly is the entire brain, because if you're talking about 10 seconds or even 2 seconds, then every neuron everywhere in the brain fires a little bit. So that's not a good idea. Second is the big difference is that assemblies that are connected already. In this world, in my world, I would say, it doesn't matter whether the neuron that fired is connected with the partner neuron or not. The only thing that matters is that in this time window, it sends an action potential. So it's very flexible. It's a very dynamic things. Now, when you have the gamma cycles, that maybe you can put together 2 or 3 or 4 gamma cycles, and this is exactly what happens in a theta cycle, because the theta cycles can concatenate, or in the neocortex, depending on you have a 4 hertz the cycles, they can concatenate the gamma cycles into successive ones, and then it looks like that you have a preconfigured ability for a syntactical operation that is absolutely needed for any kind of communication systems. So brain rhythms probably serve this very important function, is to allow to give a time frame, at least the phonetic part of syntaxis. The content, of course, is not enough, because then we have to look at the spikes and so on. But whenever you record from an EEG or anywhere in the brain, the LFP gives you a very important timing information. It says it's very important and interesting, because many neurons coalesce and they come together for a purpose. We don't know what they do. I don't know the messages. We don't know the content, but we know exactly when it happens.
So with your thinking and around how these brain rhythms work, so if I just take any everyday example, so I'm sitting here right now, and I have one visual scene. Everything that's in my visual scene right now, there's many components to it. I can see you on the screen, and I have a concept of you. I can pick you out as one entity in my visual scene. I can also see my laptop here. That is another separate entity in my visual scene. And yet both of them are stitched together into one overall visual scene. When you think about these brain rhythms and gamma oscillations and things, how is the brain utilizing these rhythms to say, give me one visual scene that's fully integrated, and yet I've got distinct entities like György and my laptop here embedded in that one scene, even though I can distinguish between them?
Okay, so if you look at the analogy to a camera, then you can say, oh, you've got a camera and it detects everything and it sees everything. If you look at from the brain point of view, or from even just the retina point of view, the reality is that you don't see things around you. Your foveal vision is 30 degrees. It's 30 degrees, okay? And it scans the environment sequentially. And you have microsaccades and so on. And the brain is also a sequential activator. So there are many, many gamma cycles that are needed to concatenate this. And somebody or some mechanism, such as a slow integrator, has to bring this together. I don't pretend I know the answer. Is that how the brain separates György from the other elements? But there has to be something like this, a mechanism that allows that, now things came together. This is the entity. Let's keep it. Let's repeat it. Let's bring it to the surface of consciousness. and then we have a understanding of what happens, and then let's move this whole thing to the next cycle, but it takes time. That's why the brain is so slow. So when in order to recognize Nick, for me, I have to say, oh, I see a scene and there is something moving. That's a very short 80 millisecond time scale. And I said, well, something that's moving is actually living. That's another 20 millisecond or so. That's a human, another 20 millisecond or so. That's a male, has beard, blah, blah, blah. And arriving to your name is about 300 milliseconds, sometimes 600 milliseconds. So there is a whole long computation slowly evolving because it requires to include more and more computational resources from a larger and larger areas of the brain, and without which the details will not emerge. So it's not a snapshot. It is well, depending on what is your view. Is a 600 millisecond is short for you? Okay,
Yeah,
it's an snapshot.
it takes time. It's an extended process.
Exactly. Exactly.
I want to ask you about so I'm going to give you the question, but then I'm going to unpack it a little bit. So I want to ask you from your perspective as a hippocampal brain rhythm guy, what memory is or how it's instantiated by the brain. And so before I let you take the reins on your answer to that question, sort of the typical way that someone who starts studying neurobiology typically is educated or starts thinking about memory is in terms of synaptic strength and patterns of synaptic connectivity. They think of, oh, okay, a memory is one set of neurons connected up to each other in a particular way. And it has to do with their synaptic connectivity and how strong those synapses are. From your point of view, what is memory and how do you think it's stored in the brain? Is it in the synapses or is it something a little bit different?
Oh, yeah. I wish I knew the answer. I don't think anybody knows. Yes, the synapses have to do something with it because those are plastic.
Mm-hmm.
Channel distributions are also plastic. There are many things that are plastic in the brain. However, if you look at if you follow the recent literature, again, neuroscience and science is cyclic, so ideas come and go. So the idea of engram was dominating the thinking for many years ago, and now it came back. It's very popular, which is basically the definition, which is there's no good definition of engram, but the old definition of this is like a picture. It has an entity, a physical entity that I can extract, manipulate, transplant, implant, hold in my hand, and give it to you. So 50 years ago, there was a big wave in neuroscience. I was a beginner at the time, but I remember the details. This was the memory transfer experiments that started with planaria. One group of animals was eating up the brain of the trained animals. And then it was transferred to mammals that they learned something in darkness and their brain were extracted and they synthesized something called scotophorbin. And then the scotophorbin was injected into the other animal and the other animal without learning learned it. Now, the modern day replication of this is the engram, which is you label neurons during learning. It's a wonderful technique. And then you reactivate them in a different context. And then voila, the animal behaves as if it was in their original environment. Now, with all my respect, I cannot imagine how this happens other than the experimental design is not proper. Most of these experiments are done in field conditioning paradigm when the only repertoire of the animal is freeze or not to freeze. And this is the measurement. And many things can lead to one or the other. Now, the reason why it is difficult to believe is because of what I just mentioned you, that there is a log normal distribution of neurons. There is another thing is that things evolve in time. And it does matter whether you have neuron A, B, C, D, or just in any configuration, and we and others have very nice support for that, or you just blast the neurons, and then all of a sudden the behavior comes in. So this is the strongest evidence for an engram. Now, it doesn't mean that rejecting the engram, I can give you a better explanation. They said, oh, there's sequences of neurons, and then if you want to have a metaphor, memory is like a cloud, the sky. It represents something, but the configuration is different all the time. And some resemble one and the other, and you can make pictures of the sky and say, oh, this is something like, and you pull out the most probable thing under the conditions where the brain is. So the brain state is very strongly determines what you pull out from the brain. If I ask you something about, what is the second largest town of Hungary, you have no clue whatsoever, and you know immediately that you don't know the answer. Mm-hmm. But if I ask you, what is the name of this photograph, then you say, I should know. This is called knowing of knowing. You know it's in your brain. You just
but I can't find it yet.
You can't find it yet. So you have to need a sharp wave or two to bring it to the surface of consciousness, and then that thing comes up, but it's not exactly the same every single time. It's modified all the time. Memories are modifiable. Every single time it comes to the conscious level, it is put into the present context, and when we put it back into long-term memory, it goes down with its modified version, as a modified version. So that's why I said it's almost like a cloud. And if it's a cloud, then the question is how redundant it is. Is it, again, that you need 500 synapses, you need 5 million synapses, or 50 million synapses to get a clear memory? And how efficient is the brain, or how efficient the brain would be? So people have done calculations along these lines. You can see that I'm not answering your question correct, targetedly, but I'm repeating what has been done. People calculated that how many synapses needed to be involved, and then how soon those synapses will overlap and saturate, and we reach catastrophic interference and so on and so on. The truth is that my brain doesn't have catastrophic interference ever. Mm-hmm. And the answer for that is because it already has preconfigured sequences. I have enough sequences forever for the entire time, even if I would be as smart as many others and Einstein, my number of sequences would not increase. It would remain the same. Otherwise, the stability of my brain would change. So if you have the sequences, then you can think about is if the number of synapses matter, then it's very limited. The number of accords you can play on the keys with the keys on the piano, you can calculate it exactly how many. But if you give time and you allow melodies to be played, that is sequential activity, then the repertoire is unlimited. Mm-hmm. And this is something like how I think that the memories are about. That is,
so you have a limited number of notes, a limited number of chords, but if you enable them to be sequenced together and concatenated across time, you can take something that's highly finite and very workable, just a few notes, a few chords can turn into an almost infinite number of songs.
Exactly. This is like humans and the language syntax. Mm-hmm. 32 letters, we can describe the knowledge of humankind. And probably that's the case also in the brain, that the brain rhythms allow you the concatenation. And because of the concatenation rule, the sequences can be much, much, much larger than frozen time-independent entities, which are today is called engram. Mm-hmm. Mm-hmm.
Interesting. So you're saying, I think what you're saying around memory is you think, and we can have some confidence perhaps, that memory can't merely be a set of synaptic configurations. The synapses are probably involved, but it can't
not a
just be
frozen,
that. Yeah,
not a frozen,
not
you know.
frozen. I want to talk a little bit more in the little bit that we have left here about brain rhythms and just what the brain is doing generally and metabolism. I'm wondering if we could we'll talk about one of your papers here that maybe ties this together. You have a paper that caught my attention. I have to confess, I haven't sat down and read the whole thing yet. But you have a paper that links hippocampal sharp wave ripples, this particular type of rhythm that emerges from the hippocampus, to glucose homeostasis. So literally blood sugar levels in someone's blood or in an animal's bloodstream. What is the relationship there? And why would a brain rhythm from a structure like this tie to a simple metabolic thing in the body like blood glucose homeostasis?
Well, this question brings us back to Ramon y Cajal when we started that
Yeah,
the wrong
yeah.
gave us a picture. But in fact, what happened is that after the wrong anatomy, when Max Cowan and others figured out that the main projection out of the hippocampus is back to the neocortex, then everybody has been obsessed about, including us, mostly us, but everybody else, is about hippocampal neocortical communication, because
Mm-hmm.
that's the fascinating part about memory. But we forget about the downstream. And then when you look at, as I said, how this wiring came up, and you look at simple organism, you can see that the sharp wave ripple is already there in organisms that have zero or very little real neocortex.
Mm.
So it had to be the case that probably that pattern emerged in evolution to serve something useful, and that's serving the body. Now, the sequential activity in the brain is already an inheritance from the body. The body is in order to do things, it has to be sequentially organized, even in animals that don't have a brain.
Yeah.
So that is, how we talk about homeostasis and things like that, but the real thing we have to talk about is allostasis, or this how from the it's not only a feedback, it's also a prediction, what I have to prepare my organism to confront something that is coming up. And that kind of thing is probably what was served by the sharp wave ripple, is that it has to change things via the lateral septum and hypothalamus. And then, as it happens many times in evolution, a good working function is accepted,
Yeah. Co-opted for another use.
and it co-opted for another use, and that became very useful because the pattern was already there. It did the little packaging, and it had the powerful burst. The sharp wave ripple is the most powerful synchronous pattern in the entire brain.
What is it? Can you describe it concretely for people? What kind of frequency are we talking about here?
This is a short 100 to 140, sometimes a little bit longer, but typically 100 millisecond time window, 200, let's say 120, 180 hertz oscillation. It's not high frequency, it's not megahertz, but compared to all the others, it gives you 5 millisecond packages. And that's probably one thing, but the other important thing is that why we are having this discussion, the number of neurons that happen in a 100 millisecond time window in the hippocampus, the neocortex, is at the order of 0.1 to 1% of the neurons. So 1% of your neurons are firing together now in a time window of 100 milliseconds. When it comes to sharp wave ripples, it can be anywhere between 15 to 42%. So it's an extraordinary strong gain of excitation. It's like sending out a shout to
Yeah.
and this is why,
And,
Nikos Logothetis and people who looked at the entire brain, you can see that the entire brain virtually is affected by this sharp wave ripple.
and if that many neurons are active, that's a lot of ATP will be needed.
Well, wait a second. No. A lot of neurons firing together is not more costly than the same number of neurons firing not together. Okay? So the interesting thing about synchrony, that is the cheapest way to make a big bang.
Yeah, it's more economical than not
It's
having synchronous activity, but it's still in absolute terms quite a lot of energy consumption. Yes.
well, if look at what happens when 100 arrows are released at the same time,
Mhm.
the impact is large as opposed to one by one.
Yes.
But the energy cost remains the same.
I see. Okay. Yeah, you get
That's
more bang for your buck, is all you're saying.
exactly.
Yeah.
That's synchronization and oscillation. That's the cheapest way of coordinating activity in time with virtually no cost.
Mhm. Now, so what is the
In,
effect of the sharp wave
in
ripple
fact,
from the
that was
hippocampus
the
on blood glucose? What is the actual result that you guys found?
so the actual result is that every single, on average, every single sharp wave ripple, the blood glucose level drops. And the larger the number of sharp wave ripples in a time window, the larger the drop. And why is it interesting? The reason why it is interesting is that type 2 diabetes, obesity, all these things have been associated, and many psychiatric diseases, they are associated with problems in sleep.
Mm-hmm.
So the thinking is that, yeah, you are sick, you are obese, therefore you don't sleep well. But the reality is that you can think about differently, said, well, something happened in the brain that changes the sleep pattern,
Yeah.
that changes sharp waves. In every single psychiatric disease model, sharp wave ripples are altered. So you change the brain pattern, and that altered brain pattern during sleep is instrumental or perhaps causal to the daytime activity. And therefore, if you don't have good sleep, you may have a high risk of
Mm-hmm.
obesity or type 2
Mm-hmm.
diabetes.
Mm-hmm. So I think what you're starting to get to here is we know that there's a correlation, there's an intimate relationship between all of these metabolic issues and chronic diseases and disruptions of the brain of different kinds. And people often tend to think, you become obese or metabolically disrupted, and then that disrupts what your brain is doing. But it could be the case that your your brain is not sending out all of the rhythms and ripples like it should be because you're staying up late to work a third shift or you're looking at a screen with a light and it's just disrupting all of your circadian biology and stuff. And that has a downstream consequence for the rest of the body, perhaps.
Exactly. So we address this by artificially making ripples, and we showed that is sufficient to decrease the glucose level. So that's causal, if you want to use this word code.
Yeah, yeah. And when and how do sharp wave ripples like this normally arise in a natural condition for an animal?
So they cannot be triggered, they cannot be induced. They, what I call, emerge.
Hmm.
And they emerge at the time when the hippocampus is losing its grip from the subcortical neuromodulators. Any small amount of acetylcholine kills the ripple. Any small amount of norepinephrine kills the ripple. It's because all these neuromodulators work on potassium currents and so on. They prevent synchronization. They prevent working togetherness or working together of the whole point of these subcortical neuromodulators that they try to make independent uh, computations as much as possible. So if you look at a isolated hippocampus, in my previous slide, we transplanted embryonic hippocampus to the adult hippocampus in either isolation or into the hippocampus itself. And we showed that the hippocampus in isolation does nothing else, just sharp wave, nothing, sharp wave, nothing, sharp
It's
wave,
like
nothing.
it's like a default.
It's a default. And that's exactly what happens in the neocortex. The default is up and down state, up and down state, up and down state, and neurons burst, pause, burst, pause.
Okay.
They finally get in
So
order
this is
to
why non-REM sleep looks the way it does on an EEG. In the absence of all those neuromodulators, you get this slow oscillation of up and down states.
exactly. So in order to take out the neurons from that enslavement, then you need subcortical neuromodulators. So when the animal is stopping, because the animal's goal is to reach something. It's always the brain generates actions towards a goal. Once the goal is reached, the animal stops, eats, drinks. It's called consumatory states. At that time, subcortical neuromodulators drop and allow the emergence of sharp wave ripples.
So sharp wave ripples are not only happening during certain phases of sleep. They can happen during waking when the animal is in a consumatory state.
Exactly. Exactly.
So when you've reached a goal, you've gotten the prize, the food, the water, the comfortable bed. That is the context in which the sharp wave ripples normally come about.
Yes. And these waking sharp waves, we just had a paper about this recently, these waking sharp waves mark or tag the important aspect, what led to this result or led to the success for future consolidation that comes a little bit later during non-REM sleep.
Yeah.
So every single night, humans have about 2,000 to 4,000 ripples. So you have 2,000 to 4,000 chances of consolidating snippets of the waking information.
Is that mostly during non-REM sleep or a certain part of sleep?
Exclusively during non-REM sleep, compressed mostly during deep stage
The
4
deepest
sleep.
phases. So people who have a disrupted sleep pattern or circadian biology are going to not be going to that deep phase and therefore not getting the sharp wave ripples. And that's there's a clear line you can draw from that to the glucose result and how it might connect to metabolic dysfunction.
Not only them, including me, older persons, we don't have stage 4 sleep with the old terminology. You don't have slow wave sleep, and the number of ripples are fewer. The growth hormones are not released properly, and so on. So we are much more prone to metabolic challenges.
And there's no good way to reinvigorate that sharp wave ripple tendency.
Yeah, of course, there is a way, and we have had a paper about that. We can prolong sharp wave ripples, and we showed that the cognitive ability of the animal increased.
How do you do that, though?
So what you can do is optogenetically, you can activate neurons at the time when a sharp wave occurs. It's a closed loop stimulation system. You put in 6 micro LEDs into the left and right hippocampus, and whenever a sharp wave occurs during
so
learning,
a scientist can invasively induce them by doing that kind of experiment, but is there a way to do it?
Now we have to go back to the hierarchy. So the sharp wave ripples are entrained by sleep spindles, the phase of the sleep spindles, which in turn are phased by the up and down states. And you can force the up and down states by stimulating the brain or the cortex by some means, either acoustically or transcranially. Some people had success with it, others haven't. Jan Born had a fantastic paper about this, showing that in healthy German students, how insight was increased by this manipulation. And he attributed it to sharp wave ripple entrainment by neocortical activity. So indirectly, you can affect this.
But we've covered a lot already.
There is no recipe for that. I have to be very clear about it.
Yeah, there's no recipe for it, but obviously this would definitely be one more reason why keeping a healthy sort of sleep and circadian biology schedule is critical.
Please advertise that.
What are some of the things that you're working on today in the lab that you're particularly excited about that we can look forward to coming out in the next couple of years?
Is the same thing, brain-body problems. We are trying to get more and more information in today's neuroscience, because in today's neuroscience, you have to find your own niche. And I think most neuroscience today goes towards the path of least resistance of technology. And we keep finding the same things over and over again, shown better and better, because we use preparations for convenience. What I mean here is the head fix preparations became so convenient and it's everywhere that people who would like to understand about the contribution of the body and the vegetative nervous system, they lose track of it because they don't see the key things. So this is what we try to exploit, that everything should be freely moving, more ecologically defined environments, recording from the heart rate, recording from whatever you can. And there are past measurements available, not only for glucose, but for other other substances in the body that have been never considered seriously. If you look at these pyramidal cells in the neocortex, you will find that they have receptors for, let's say, 30 known ligands, but there are many, many, many more that are available to be modulated by other things, such as body circulating substances. at a much slower time scale, and that hasn't been looked at. And the reason why I'm interested in this and take it seriously is because of my extension of the hierarchy of the brain rhythms to the brain body rhythms. So I see it as one entity. This, of course, is not only my lab. There are many labs that are going in this direction, and there are technological developments across the US and the world that makes it happen. But I think that's the next stage that you should be looking at.
Are there any final thoughts you want to leave people with, especially people in and adjacent to neuroscience, in terms of how they might want to reflect or reconsider or examine some of the assumptions around how people often think about the brain?
Well, given the current situation, I think I have only a short-term message, is that I see the high anxiety among my students and postdocs about what's happening next. And I think is that the brain has this log rule that the longer you lived, the larger epochs you see, and they average out. So just a couple of years of upheaval shouldn't make you change your mind if you wanted to become a neuroscientist, because it will even out. In a couple of years, the situation will be much better. I hope it's a positive comment, and I don't want to, guide young people into the wrong direction. But if you are fascinated by science, then stay in science. I don't have any recipe. Nobody has recipes. Neuroscience is the Wild West. Any person can have the same chance of discovering something big, almost, than the Allen Institute, because there is so much to learn. One sentence that we use in the lab all the time is that it's impossible to find nothing in the brain.
All right. Well, Professor Gyorgy Buzsaki, thank you very much for your time.
Good talking to you, Nick. Take care. Bye.
Visit mindandmatter.substack.com to find all of my content. This episode is supported in part by Athletic Greens. Their main product, AG1, is a comprehensive and convenient daily nutrition product containing 75 vitamins, minerals, and whole food sourced ingredients with less than 1 gram of sugar per serving, no nasty chemicals or artificial anything. It's gluten and dairy free and compatible with paleo, vegan, vegetarian, and ketogenic diets. AG1 is a quick and convenient way to supplement your diet to help ensure your body is getting the nutrients it needs. It comes in powder form, and you can mix it in water and drink it, or you can put it into a smoothie or a shake or something like that. I mix it into water and drink it, and it's super convenient. If you go to athleticgreens.com/mindandmatter, Athletic Greens will give you a free 1-year supply of vitamin D and 5 free travel packs with your first purchase. Their vitamin D product comes in tincture form, so you just take 1 drop each day. A large fraction of the population is actually vitamin D deficient, especially in winter months when we get less sun exposure. And vitamin D is super important for the proper function of the immune system and for a variety of other things. So visit athleticgreens.com/mindandmatter or click the link in the episode description. You'll get a free 1-year supply of vitamin D with your first purchase.